Hello, and welcome to our webinar. Thank you very much for joining us today. My name is Jan Nechamen. I'm the immunoassay platform specialist for Bristol Squibb, and I would like to speak with you today about the multiplex immuno-oncology panels 1 and 2. Our general menu of kits or portfolio, if you would like, contains just about every single species that we generally study, including ovine. Within those species, we study all different types of disease models, whatever may be of interest to every different group. This generally comes together to roughly 230 kits and about 1,200 analytes, and this portfolio is constantly growing. We are happy to announce that we now have a human metabolic panel 3, but that's another conversation. Today, however, we're talking specifically about cancer and the 10 hallmarks of cancer. We know that cells are generally under a tight control, and if they get out of this control, our body is supposed to either put them back or eliminate them altogether. We also know that a lot of cells get clever and actually find ways to fool the immune system and escape general control, which leads to cancer as we know it. This is actually done through cell-to-cell interaction, or specifically cell-bound proteins interact with the receptors on the T and B cells. And that's not without help of generally soluble cytokines and chemokines, but these are key factors. This brought us to create a first panel of checkpoint inhibitor panel, specifically because we learned that these molecules can be cleaved and they can float around freely in the matrix. The matrix, of course, being serum or plasma. This panel was looking at the most popular things to date. Some of the molecules are like CD80, 86. We know them as costimulatory. Some of the molecules are PD1, PDL1, PDL2, which we know are inhibitory. I'd like to note that PDL1 and PDL2, although have homologous structures, our antibodies are specific, do not cross-react, do not interact, and the data is actually spot on. It's been tested and verified. To bring your attention to some of the information about the kit, here are the representative standard curves, and I would like to point out specifically the precision of the assay, or the intra and inter-assay precision, contains CVs that are generally 5 or 10%. Only 1 or 2 actually have a CV of 15%, which is well within the norm of industry standards. Well, what do we get out of having a panel like this? As an example, we ran 3 breast cancer cell lines and 3 colon cancer cell lines. And we can see that each one of them presents a slightly different fingerprint, if you will, or profile of the different molecules within this panel. And that can actually give us a little clue about perhaps the stages of the disease or how to approach treating these diseases. Recently, we generated panel 2, which is actually just about 2 months on the market. This is a 31-plex panel that contains things like an expanded B7 group of markers, BAF, or as we also know it, BLIS, MAC-A, MAC-B, 4-1BB ligand, and X40. So a slightly more expanded picture of what is actually going on with our tumor microenvironment. Here, too, I would like to present you the representative standard curves, and I would also here like to point out specifically that the intra and interassay specificity is super tight at about 5 or 10%. Just like the previous panel, this is run on a breast cancer line. This is just an example, but here we could see CD40 ligand, TIM3, GAL1 are expressing at significantly higher levels as cleaved molecules, as opposed to, for example, CD40 ligand, which seems to be still stuck on the membrane. This allows us to see a certain picture of the tumor microenvironment, of the tumor disease progression, if you will. And that's only part of the story. And our kits are always built to be complementary to each other. So if you're studying Th17 response, CD8 response, or maybe looking at soluble cytokine receptors, we have panels that will help you complete your picture. And these are not the only panels that we have. Like I said, our menu is large, so you could look at other inflammatory immunology panels to round out the picture and to tell the true story about what is going on in this disease state. And with that, I'm Very grateful for you for your time, and I would like to turn over to Jenny, who will tell you how these panels are used within her laboratory to show you how these panels benefit her studies.
Thank you very much, Jan, for the kind introduction. I am going to share with you how we use high-throughput biomarker profiling in immuno-oncology research and development at BMS. I will introduce our biomarker platform and the role biomarkers play in early-stage IO discovery. I also want to share our experiences with the multiplex assay, as well as show you some case studies of how we apply them. While most of my presentation is focused on IO discovery applications, I want to take a small detour into biomarker profiling for cardiovascular disease. Finally, I will give you an outlook of our future plans for biomarker profiling. The group, based in New Jersey, serves as the central biomarker profiling hub for all discovery needs. We routinely work with and receive samples from our US research sites in Massachusetts, California, and Washington. We also work with international collaborators around the world. In order to facilitate high-throughput analysis, we have established an automated and miniaturized process workflow that is based on the XMAP technology, which, as most of you will be familiar with, is an antibody-based immunoassay. The capture antibodies are coded onto color-coded beads, and the detection antibody signal is quantified via a biotin-streptavidin PE interaction, which ultimately allows reading out a fluorescent intensity signal that scales with the abundance of the analyte in the sample. Standard curves serve to interpolate the absolute amount of each analyte in the sample. We run all samples in a custom in-house developed 384-well process, which facilitates a 75% cost saving and increases sample capacity fourfold, while also reducing the sample volume needed significantly. In this process, we make heavy use of a number of automation platforms, including liquid handlers and workstations. For example, to support the consolidation of samples from submitted sample plates into master assay plates. We have a dedicated fleet of readers with integrated automation solutions for loading and unloading plates that can run pretty much 24/7 to process the data. Finally, our entire process is critically dependent on a custom internal IT platform, which allows our customers to submit their profiling requests online, helps us with reagent inventory and study data management, data analysis and QC, and supports the generation of customized reports for the different research communities that we partner with. The team currently runs biomarker panels for 5 species: human, mouse, rat, dog, and cyno. And we support projects from the early exploratory to the clinical phases. The reel on the right-hand side is intended to give you a flavor for the types of studies that we routinely process. One category encompasses mechanism of action studies, where we profile samples generated from cell cultures, for example, co-cultures of tumors and immune cells, or ex vivo stimulated preparations of primary immune cells and whole blood. Another big focus area are PK, PD, and efficacy studies in preclinical animal models, such as syngeneic or genetically engineered tumor mouse models, but also rodent models for fibrosis, cardiovascular, or autoimmune disease. A third and ever-increasing category of our work falls into the realm of toxicity studies, including exploratory tox studies for investigational new drug applications. Things we often look for with the appropriate Analyte panels, for example, include cytokine storms, immune activation, as well as liver and kidney injury. The last category branches into the areas of exploratory biomarker identification and patient stratification, where we profile biobank samples, including those from internal clinical trials, as well as various disease population samples that we typically source with the help from our academic collaboration partners, including the corresponding normal healthy volunteer controls. In order to be able to support this wide range of study types, we need a very broad coverage of Analyte panels for the different purposes and therapeutic areas. We currently have over 600 Analytes in active use. Around 60% are from a variety of millipores multiplex panels. We also have an ever-growing list of new Analytes in development, which can either mean that we are working to miniaturize existing panels to add to our capabilities, or develop completely new Analytes, mostly through very fruitful collaboration with the millipore team. Our different panels are broadly classified into innate and adaptive immunity, heart failure, fibrosis, and toxicity. The immunology and the soluble checkpoint panels are the ones I will be focusing on for the remainder of the webinar, with a small excursion into our cardiovascular and heart failure Analyte applications. I had mentioned previously that a lot of our remit and biomarker profiling at BMS is on high throughput. And as such, we have optimized the workflow to be able to handle up to 2,000 samples every week, with sample requirements as low as 5 microliters per panel. A large fraction of the sample types we deal with are serum, plasma, or supernatant or lysate from a cell culture or tissue. We do, however, also work with more exotic matrices, such as azides, blister fluid, synovial fluid, bronchoalveolar lavage, or increasingly urine. We have been building out this capability in the last few years, and its use across the BMS research community has really taken off. To date, we have profiled more than 250,000 samples for more than 170 different programs, which has resulted in more than 11 million data points. That brings its own challenges in data management, which is why the custom in-house IT platform I mentioned previously is of crucial importance. Now I want to switch gears a little bit and focus specifically on our immuno-oncology support. Of the roughly 47,000 samples we analyzed last year, almost half came from IO. As you can see in the breakdown, the majority of samples are from human origin, followed by mouse and cyno. On average, each individual study has 70 samples, but the range per study is very wide, from as little as 10 to as many as 800 individual samples. Similarly, we profile a mean of 41 cytokines per sample, across a range from 14, which would be a single panel, up all the way to 98, and multiple panels being run for broader profiling. The average number of panels we run per sample is 3. We have certainly seen a demand for more deep profiling, especially for precious sample, in line with more and more analytes covering different biological pathways becoming available. Now, you might ask yourself why we expend such a large effort to investigate soluble biomarkers in general and cytokines in particular for immuno-oncology research. It might become clearer when looking at cancer as essentially the result of a dysregulated immune system, as in Ira Mellman's and Dan Chan's cancer immunity cycle, where every step of successful tumor eradication is dependent on cytokines as master regulators, from tumor antigen presentation to the activation and priming of relevant immune cells, trafficking of those cells to the site of malignancy, and finally infiltration, recognition, and destruction of the tumor cells. We have long known that cytokines play dual roles. They contribute to tumorigenesis, for example, by promoting pathogenic inflammation and tissue damage or invasion, metastases, and angiogenesis, but also play key roles in preventing tumor formation or resolving early-stage malignancies. Understanding this intricate balance of the immune system's master regulators is key in developing successful IO drugs. And the first step is understanding which molecules are present at what stage of cancer development. Cytokines themselves are in fact also interesting therapeutic options for cancer treatment. This graphic from a 2019 review shows which ones have been or are currently being investigated in clinical trials, from interferons to GM-CSF and various interleukins. Interferon and IL-2 are approved treatments for hairy cell leukemia and melanoma and renal cell cancer, respectively. Both mechanisms of action rely on broad immune stimulation. With this outsized role of soluble biomarkers, including cytokines, chemokines, and soluble checkpoint molecules, it is probably not surprising that we have a large selection of analytes in routine use for IO-focused projects. Here, I am showing the 133 analytes and their corresponding panels that we use for human sample profiling. We have similar, if somewhat smaller, selections we use for mouse and cyno studies. All these panels I'm showing here are based off existing multiplex panels, and I want to use the next couple slides to show some of our experiences working with those reagents. As we are running so many different samples and analytes, we need very robust kits and reagents. Generally, we require strong performance when it comes to reproducibility and precision, sensitivity, and specificity. Reproducibility between assays, but also between different lots of kits, is key as we support multiple longitudinal studies where samples can be received months or years apart. In addition, a good performance in our custom 384-well format is essential. This is something we will extensively test when new multiplexes come on the market that we want to add to our platform. Additionally, we work with an internal team of statisticians to ensure a robust QC process for all runs and reliably determine the lower and upper limits of detection and quantification. We have a database that keeps track of the key performance indicators for each assay run that will automatically flag if data falls outside of the pre-specified acceptance criteria. For the aspects of sensitivity and specificity, I will share some observations in the following slides. Sensitivity, and in particular the lower limit of quantification, are critical aspects for a lot of cytokine biomarkers, as a large fraction of those proteins are low abundance, particularly when using unstimulated samples or healthy controls. We have frequently struggled with detecting certain biomarkers reliably in different sample types, let alone properly quantify them. That's why we are always interested in new high sensitivity kits. The data on this slide shows our experience with the high sensitivity multiplex panels for human and mouse. We are comparing the LLOQ, the lower limit of quantification, between the original panel and the high sensitivity version. Note that LLOQ values are determined from the performance and fit of the standard curve under our specific 384-well assay conditions, and our protocol differs from what comes with the kit, so your individual mileage may vary. As you can see, most of the analytes have an improved LLOQ, in some cases very significantly, by 50 or more fold. Some analytes for this has been especially helpful are IL-10, IL-12P70, IL-4, IL-5, and IL-6. For our customers, we generally recommend the high sensitivity panels as the default choice when multiple panel versions are available, and aim to move as many analytes into those types of multiplexes as possible. Next, I want to give a little insight into specificity using the soluble checkpoint panels, which are of key importance for IO drug research, especially when it comes to patient stratification. The data on this slide refers to the 17-plex checkpoint panel 1 that we adopted a couple of years ago. When we initially investigated available panels in the market, we wanted to get a sense for how they performed against qualified recombinant proteins. Owing to our broad project portfolio, we had a variety of in-house produced recombinant proteins available that we used for this purpose. The heat map on this slide shows the fluorescence intensity values across the 17-plex on the x-axis versus the kit's standards and our spiked-in control proteins PD1, PDL1, PDL2, and IDO on the y-axis. Across the top, you can see that each standard produces a nice dose-dependent signal in its respective channel. Going down the list, you'll notice that in our hands, the specificity of the kit is excellent. Each recombinant protein only produces a signal in the channel or with the bead we would expect it in, and everything else looks like background. At the time, we were also evaluating other kits, and I'm showing an example that didn't quite behave as well in this slide. Same concept as before, slightly different analyte composition. The kit standards once again behave well, and the control proteins produce strong signals with their respective beads, as you want to see. However, under our assay conditions, with this kit, we also observed significant cross-reactivity with several beads, which means a signal is showing up in a channel where you would not expect it. See the blue arrows, which ultimately means that we couldn't be confident this particular multiplex would faithfully detect the independent checkpoint analytes in our patient samples. Next, I want to share with you some very exciting data from a new 31-plex, the Millipore Checkpoint Panel 2, where we were involved in the beta test process. This panel is an exciting extension as it contains a broad variety of additional checkpoints and ligands, such as members of the B7 family, and many of the analytes are complementary to Checkpoint Panel 1. As before, in this beta test, we ran a variety of recombinant proteins against the multiplex and see very good overall specificity. We have since begun testing this panel on real-world samples and see similar encouraging data. I believe this panel has now also launched and reagents are commercially available. So we are working to add it to our platform for routine application. Now, for the next section of my talk, I want to focus on some key applications for biomarker profiling in our IO discovery research and share some case studies to give you a flavor of how we use the data to progress projects. The case studies encompass 3 different flavors of experiments. First are in vitro or ex vivo cell models, such as profiling the biomarker signatures of primary immune or tumor cells, as well as co-cultures. We use these experiments typically early in a project during the target validation phase. The second type of experiments are in the preclinical or in vivo group, where we profile tumor models in animals, either in vivo or ex vivo, and also analyze samples from exploratory tox studies, often performed in cynomolgus monkeys for a particular drug candidate. The third category encompasses all types of patient samples. These are typically the samples that are the most precious and where we perform the broadest possible analysis. In the early stages of an IO discovery project, we use a number of complex cellular models to profile a particular target's or compound's behavior. One of those models is the Coculture of genetically engineered primary T cells, which express a known antigen-specific T cell receptor with matched tumor cells presenting the cognate antigen on their MHC molecules. We can measure tumor cell killing as shown in the pictures on the right. In the absence of antigen recognition, the tumor cells in red will keep growing. In the presence of the antigen, the T cell activates and lyses the tumor cells, as shown by the reduction of the red signal and the increase of the green annexin 5 positive clusters of dead tumor cells and T cells. T cell activation results in a very strong increase in cytokine production, so we frequently use the milliplex panels to quantify those changes. Specifically, we like to use the 17-plex that includes key T cell cytotoxic markers like granzyme and perforin. An example using the Coculture system I showed you on the last slide is depicted here. The idea of this particular project is to turn cold tumors without antigen presentation into hot tumors that do present antigen, using various types of epigenetic modulators that we treat the tumor cells with prior to adding the antigen-specific T cells and measuring their activation. The heat map shows the cytokine data from the 17-plex for a selection of key analytes. Data is represented as a log two-fold change of the induced cytokines over the control in the absence of antigen-specific recognition. Across the x-axis, we have a selection of tumor cells. The wild-type tumor cell that is loaded with the antigen serves as a control. And you can see that the presence of the antigen added to the cells externally strongly induces T cell activation and cytokine production. This data also shows that the first epigenetic modulator that we tried does indeed induce the tumor cells to begin expressing and presenting antigen, which in turn leads to measurable T cell activation. The second molecule tested does not have the same effect. We wanted to make sure that this observed behavior was indeed due to the tumor cell antigen's presentation pathway, which needs a whole machinery from gene expression to protein production and processing of those proteins through the proteasome to the eventual loading of the antigenic fragments on the cellular MHC molecules. Therefore, we created some CRISPR knockouts of key proteins in those pathways. The first one is a beta-2M KO. Beta-2M is an essential component of the MHC complex on the cell surface. So in the absence of this protein, no functional MHC complex can be formed, and the cell is not capable of presenting any antigen, irrespective of whether they are derived from inside or outside of the cell. The second knockout is a TAP1 KO. TAP1 is an essential transporter for intracellular antigen processing and loading. So in the absence of this protein, the cells are expected not to be able to present newly expressed neoantigens, but are anticipated to have functional MHC complexes on the cell surface. Indeed, our cytokine data using the antigen-loaded controls confirm these expectations. The tumor cell without a functional MHC cannot induce T cell activation even in the presence of the antigen, whereas the TAP1 KO that still has the MHC complex is performing like the wild type. In both cases, our epigenetic modulators don't work anymore due to the deficiencies in the intracellular antigen presentation pathway, which, together with other data not shown here, gave us confidence that the approach of epigenetic modulation is promising. My next case study comes from a mouse tumor model, specifically an orthotopic transplant model of pancreatic cancer that was studied in collaborations with a group at Johns Hopkins. The specific question in the study was if radiation therapy, a standard of care approach, can enhance immunotherapy efficacy of PD-1 inhibitors or combinations of PD-1 and IDO inhibitors. Ultimately, the data revealed that survival was improved by a combination of radiation and anti-PD-1, but that IDO combinations were not useful, and that this treatment effect correlated with a systemic interferon gamma response as shown by multiplex cytokine profiling of mouse plasma samples. Now, I want to give you a flavor for how we use cytokine profiling to evaluate the toxicity risks of our later stage molecules before filing an IND. In this particular example, a BMS molecule was given in increasing doses. Group 1 is the control group. Group 2 through 4 received the drug twice a week apart. We received serum samples for cytokine profiling at different time points, from just before the first dose until about 2 weeks after the last dose. We assessed a total of 44 different cynomolgus analytes in 2 panels, I have chosen an example of 5 relevant ones for the line graphs, where each line represents an individual monkey. We observed a dose-dependent transient increase in inflammatory cytokines that peaks 3 to 6 hours after dosing and returns to baseline within about 24 hours. We also saw that the second dose did not cause the same magnitude of effect. Together with other data, we could conclude that the molecule was well tolerated and did not cause a cytokine storm in the animals, which is a concern for the particular mechanism of action we are targeting with this drug. Based on other in vitro data generated over the course of this project, we could also conclude that the observed cytokine induction is largely on target and on mechanism. In my last IO-specific case study, I want to talk very briefly about our work with patient samples. When we obtain these types of samples, we typically perform broad profiling, as much as the sample volumes permit, often for 100 plus analytes. This is also where we mostly use the checkpoint panels, as exemplified in the heat map, which aggregates several hundred serum samples by tumor type, and for which we can see clear differences in soluble checkpoint molecules detected. Ultimately, the goal is to build a sufficiently large database that allows to identify predictive biomarker signatures when linked to patient phenotypic or treatment data. Another focus for us is performing longitudinal studies on treatment, particularly with anti-PD-1 therapies, to investigate response and resistance mechanism and their impact on biomarker signatures. Further, we also use tumor explants to evaluate individual treatments and treatment combos, and the biomarker data is typically a key readout in those studies. With that, I want to take a little excursion into the realm of heart failure biomarkers and share with you a study we contributed to that had considerable translational impact. Heart failure, in particular the subtype with preserved ejection fraction, also known as HFpEF, where the left ventricle is not able to fill with blood properly during the diastolic filling phase, but otherwise appears to pump normally, represents an increasing burden on public health, especially in aging population. It has, despite many attempts, no proven pharmacological interventions, and its 5-year survival after a heart failure hospitalization event is only 35%, worse than several types of tumors. It is being increasingly recognized as a very heterogeneous disease that it has in common with cancer. No distinct biomarkers exist, and remedying that was the objective of this project we contributed profiling data for. The samples for this effort were sourced from a biobank of the TopCat clinical trial that investigated the use of the aldosterone antagonist spironolactone in HFpEF patients older than 50 in an international trial. We obtained these samples for a very active collaboration with UPenn. Ultimately, this trial, like many others before it, failed to meet its endpoint, which was a composite of death, aborted cardiac arrest, and heart failure-related hospital admission. We profiled those samples using a custom BMS cardiovascular panel that includes 50 analytes covering a multitude of biological processes, from inflammation to angiogenesis, fibrosis, cardiomyocyte and adipocyte biology, and more. In our analyses, we found that the peripheral biomarker data, together with the patient metadata, helped stratify the trial population into 3 distinct subgroups. Key circulating biomarker, cardiac, and vascular features were found, indicating distinct patterns. The phenogroups exhibited different prognoses and differential responses to spironolactone. The first group was slightly younger, with milder symptoms, a high likelihood of smoking, but low levels of natriuretic peptides, which are usually associated with worse outcomes. The second group skewed older and female, had more chronic kidney disease and atrial fibrillation, and a distinct signature of active mineral metabolism and inflammation. The third group was characterized by high levels of obesity, diabetes, and depression, and presented high biomarker levels for inflammation, especially with regards to TNF-alpha, fibrosis, and angiogenesis. This group fared the worst. While the overall TopCat clinical trial failed, in FENO Group 3, there were signals of a treatment benefit in this retrospective analysis. The study highlights the power of biomarker data stratifying patient populations in highly heterogeneous diseases to identify those subsets that have the highest likelihood of benefiting from a particular treatment approach. With the same biomarker data, our UPenn collaborators around Julio Chirinos built a machine learning model with the goal of identifying predictive signatures. Indeed, when looking at the entirety of measured markers versus the patient metadata and primary study endpoint, several markers appear to be significantly associated and clusters emerged for inflammatory, metabolism, fibrosis, and angiogenesis pathways. As I mentioned before, HFpEF doesn't have any well-defined biomarker signatures, but something called the MAGIC risk score is frequently used to evaluate risks for patients to die of heart failure at 1 and 3 years after diagnosis. This score incorporates a number of clinical parameters such as age, weight, whether someone has diabetes, COPD, takes certain medicines, and several other factors. The utility of this score varies by patient population. When using the MAGIC risk score to look at the TopCat patient population, its predictiveness is pretty weak, as indicated by a C-index of just over 0.6. A C-index of 0.5 means the same predictive value as random chance, whereas a number of 1 equates to a perfect prediction. The biomarker machine learning model performs much better than the MAGIC score with a C-index greater than 0.7 in the TopCat patient population. Interestingly, this evaluation also was true in a completely independent cohort, the Penn heart failure study. So essentially, the combination of prevalent biomarkers in the patient's periphery could predict if this patient would experience a heart failure-related incident, such as hospitalization or death. This, once sufficiently validated, could eventually be a new and easy way for classifying patients into risk groups from a single blood draw. Overall, this work has presented tantalizing evidence that a plasma biomarker signature for HFpEF is achievable and may outperform current methods for assessing risk or can be effectively combined with them. BMS and UPenn are now part of a worldwide heart failure consortium that aims to validate those findings in 13 international cohorts with more than 5,000 patients. I hope I was able to give you a flavor for how we use high-throughput multiplex biomarker profiling at BMS, and want to finish with an outlook on our current and future focus areas. An ongoing effort is in the expansion of coverage, adding new panels and analytes to the platform, or developing new analytes altogether in close collaboration with Millipore. We also have a growing need for custom disease-specific panels to support targeted biomarker studies. We also try and consolidate and merge panels wherever possible to reduce sample volume needs and overall workload. We also spend a lot of effort in 2 closely related areas, data analytics and data integration. I have shown you that we profile tens of thousands of samples a year across a wide variety of experimental designs. With that comes a strong need for a data and also, very importantly, a metadata infrastructure to facilitate the widest possible use of the results. So we constantly try and optimize our data registration and database methods. This is also key in facilitating enhanced bioinformatics methods for data analyses, as well as efforts to implement machine learning, particularly for generating disease predictive biomarker signatures. The heart failure biomarker study I showed you is a prime example of why these methods are of value. Biomarker data also becomes much more informative when it can be overlaid with other data types generated from the same sample, such as sequencing or phenotyping, for example. Lastly, an effort that we have ongoing focuses on new technologies that allow investigating the cytokine profile of a single cell. As we know, immune cells in particular show very heterogeneous responses to treatment, and marry it to the bulk biomarker data that we routinely produce from Luminex profiling. Finally, I want to thank you for your attention and acknowledge the BMS cytokine team, in particular, Melissa Yard and Nathan Cheadle, who are the engine of this platform and keep the samples and data flowing on a day-by-day basis. We are also indebted to all our BMS collaborators, both those who enable us to physically run all the instruments we need, as well as our partners in the individual therapeutic areas. I also want to acknowledge our strong partners at Millipore, particularly Jan and Shane, who we meet with frequently in non-pandemic times, as well as Brooke, who supports our ever-expanding list of requests for new analytes. Lastly, I want to express my gratitude to our UPenn collaborators, whose support is invaluable in the cardiovascular and heart failure space.